So, ladies and gentlemen, here we are today. It is going to be the beginning of sort of an intermediate time. Let's take a look at your syllabus. And on the syllabus, you will note that today I'll be talking about the special senses, and that's correct. And that on Thursday, I'll be discussing the endocrine system. Now, the special senses, your eye and your ear and smell and taste, that's going to be on your take-home exam. The take-home exam is going to cover 4 areas that I will talk a little bit about, that you've seen a little bit about, but we haven't dealt as in depth with. So the take-home exam, which is out there on Mastering, is due a week from Sunday. You've got 10 more days or so to do that exam. It's going to cover some on the eye and the ear and the special senses. It's going to cover some on the endocrine system that I'll talk a little bit about with you on Thursday. You've already seen the eye and the ear in lab. It's also going to cover the digestive system. You've covered that in lab. And it's also going to cover a little bit about the lymphatic system. And in the next lab, lab 9, we'll at least introduce the lymphatic structures to you, like the lymph nodes and the spleen. So you're going to have heard parts of the story, but not the entire story. The good news is the take-home exam is an open book, open resource. You should do really well on it. It's not timed. You don't have to do it all at once. And I would recommend don't wait till the night before it's due to open it up for the first time. Go ahead, go in there, answer the questions you know, maybe search out a couple that you don't know, close it up, come back a couple days later, do a few more questions, and maybe break it up into the 4 different areas. So there's senses, endocrine, digestive, and lymphatic. Don't wait till the night before. That exam is worth 40 points. It should be a strong score. That means that what I'm talking about this week is not going to be on your next exam. Right? It's on this take-home exam. So use this week. I mean, you're going to be listening to me right now, but you're going to be in the back of your mind saying, I don't need to really learn this perfectly because I'm going to have my book open when I take this test. So it's a little bit of a week off from pressure. You've earned it. Last week was was a tough exam with the nervous system exam. But this is also a good week for you to tackle your short paper. So in this week where we're kind of relaxed a little bit as far as testing goes, take advantage of that and knock out your short paper. Remember, that's worth 50 points. So you've got 90 points. You've got something more than an exam right here in front of you between the take-home exam and the short paper. And then you'll also at the end, remember, you'll get 30 points if you've been doing your mastering assignments. And the last lab is bonus points. So if you attend all 11 labs and you do all 11 pre-labs, you'll get bonus points there as well. So there are some nice grade enhancing opportunities coming your way. Hang in there. I know that some of you are a little bit depressed and upset by your scores right now, but there will be improvements coming. Yes, ma'am.
Is the
Final exam cumulative? No.
That's a good question.
No.
No, it's not, but that wasn't my question.
Okay.
I'll think about it.
It's okay. The take-home exam is not timed. It is not, you can go in and out all you want. Still not your answer? Not your question? Okay. Short paper?
Oh, the labs.
Oh, yeah.
If we go to all the labs, does that still count even if we didn't go to our?
If you missed a lab but made it up, that still counts. Absolutely. So if you've been to all 11 labs, only 10 count for your grades. The 11th one will count as bonus. So your 11th quiz and your 11th pre-lab turned in will count as bonus. So that's all good. Now there is an error on your syllabus while you're there. It's like vocab jumps. There's no vocab on this take-home exam. And the syllabus has an error. It like skips over vocab. So that means that vocab on exam number 5 will start with 73, right, and go through like 92, something like that. I'm not going to do any vocab today. I'll give you a little break, but we'll start with vocab next time, and that'll be on exam number 5. Exam number 5 will be entirely on the cardiovascular system. So that's going to cover blood, the heart, and blood vessels. Those are the topics of this week's lab and next week's lab. So you're going to have 2 labs covering blood, heart, and vessels, and the next entire big exam will also cover cardiovascular. That'll leave only the final exam, which is a non-cumulative exam over respiratory, urinary, and reproductive systems, and the last 2 labs cover those content, those systems. So we're in that home stretch. It doesn't seem like it's that close, but when we get to November, things seem to go really, really fast. You've got 3 weeks before we're eating turkey, right? 3 weeks and 2 days you're eating turkey, but who's counting? and then you come back from Turkey Day and you've got a lab exam, and then the week after you're in final exams. So it comes really, really fast. So hang in there. Other way of looking at it is that although we're near the end, you've only earned about 60% of your points. You've only got about 600 total points out of 1,000 in your grade book. There's still 400 more points to earn. There's 2 more exams, there's a lab exam, there's your take-home exam, there's a short paper. There are still a lot of points coming your way toward your point total. That's a good thing or that's a bad thing, right? It's a good thing if you're going to focus and do really well in those things. It's not a good story if you're going to blow off the short paper or blow off the take-home exam. So make sure that you're focusing on those things as you can. So today I'll be talking about chapter 15. This is actually a continuation of the nervous system. These are the special senses dealing with the eye and the ear and touch. And it's kind of relaxing. Let me also go back and say something though about your nervous system exam. That was by far the longest exam you're going to experience in my class. It was a long exam. I know it was. Some of you did not finish. Many of you did not finish. And I don't know of anyone who felt like they had enough time to really focus on everything the way they would want to. Now, call me crazy, but I think that I have more responsibility than just teaching you anatomy. You're going to have tests with you, like it or not, the rest of your life. Some of you are going to have big exams like the NCLEX or MCAT or GRE, exams that can take a long time. My daughter took the NCLEX today. That exam can go as long as 6 hours. The MCAT is now up to 8 hours. The GRE is 4 or 5 hours. These are long exams. And if I and every other instructor on this campus and throughout your undergraduate experience only give you 1 hour exams, you're never going to sort of build up to that mental level of concentrating for an hour, 2, 3, even 4 hours. So there's a little bit of medicine in that third exam with the nervous system, or the fourth exam. I consider it is a long exam. There's a lot of material. If you know it well, you probably finished. If you didn't know it very well, or you're a little bit of a slower test taker, you probably didn't finish it. The good news is there were 25 extra questions on the test. You could miss 25 questions and still get 100%. There were 5 bonus points just for reading the instructions. Only about 30% of the class read the instructions, because in the instructions it said bubble in A for 151. Somewhere else it said bubble in B for 152. At the very end it said bubble in E for 155. So first thing you look at, well, yay, but 2/3 of your classmates didn't see any of those bonus points because they didn't read any of the instructions, which are at the top of the pages in bold. So another little take-home lesson is make sure you always read the instructions. No matter how stressed out you are, read the instructions. There's always something important in there to glean that may assist you in the test itself. So read the instructions, work efficiently, keep that mind going. We had an hour and a half for that exam. You won't have any longer ones in this class. The cardiovascular exam will be shorter. It won't feel so rushed. The final exam won't feel rushed either. Okay. The class overall did really quite well. The median was nearly 80%. I had students who did fantastic. I had 2 scores over 100%. I had a large number of good, strong A's. I had a nice number of B's, not that many C's actually, and then too many D's and F's. So there was that, what I call the bimodal or the camel hump, the double humped curve. So I've got the students who really knew their stuff well and did well, and I had students who just were not prepared. And you know where you fall in that continuum. So I just had a conversation today with some of our tutoring center people on campus. And one of the conversations is, should we somehow require tutoring for this class? And I'll ask for your feedback at some point. But math has instituted, some of the math classes have instituted mandatory tutoring. And if you're not making an 80% or better, they're requiring you go to weekly tutoring. If you don't go to tutoring, they drop you from the class. You're not making the grade you need. If you're not going to go to tutoring, we're not going to waste the space. So you've got to go show up and do tutoring. I'm almost considering that here. Look around. I've had 1 student so far go look at their exam. Now some more of you will go look, but I don't have many students coming seeking out help. Our supplemental instruction, Janae, is not being used much at all. I don't think I've had 2 students all semester darken my doorstep for office hours. And walk-in tutoring and tutoring, they're not very busy. So I have a lot of students who are not doing that well in the course, but they're not seeking out help. And while it may be painful to be required to go to tutoring or extra help, it may be the ticket that I need to do and say, you know what, if you're not making at least an 80% on the exams or overall in the class, we're going to require you to go to tutoring. And once you start making more than 80, it's optional. But until you're making a B or better, we want you in tutoring. We think you need the extra help. Think about it. Let me know what kind of feedback you would, give me if you were the student being told you must go or we would recommend you go. Do you think that might be something that would be well received? Now, obviously, that tutoring time would have to be convenient. And as being more evening students, we would have to make sure that those tutoring opportunities were in the evening. We couldn't expect you to go to work all day and then somehow come back during the day. We'd have to make those tutoring options later in the evening. But I'm really strongly considering it just because look around. We started out with 40 students in this class. We saw a lot more on test day, but they're not coming and they're not doing well, many of them, and they're not seeking out help either. So I don't quite understand that disconnect. If you're not doing well, usually you would seek out help. And that's not happening. I'm not seeing that, right? And we all have different stories. We all have different reasons.
I'm a part of that. I don't know.
That's okay, Kim. We've talked. We've talked though, right?
I don't know why I haven't been received.
But we've had connection. You've talked. We've talked. But I have a lot of students who are not doing that well, but don't seem to be struggling. Don't seem to care. Don't seem to really wonder what they can do to be better. And
They would have to pay for the class if they want to. First,
the tutoring's free.
no, I mean this class that they have to pay for.
Is this expensive?
It's an expensive. I agree. This is 6 contact hours plus a lab fee and not cheap books. This is not an expensive ticket, right?
They have to pay for it again.
I'm going to say something off record,
I was gonna say job security.
but I can say, but they should be coming to me and saying, thank you, Dr. Macauley, for paying that for me. Because someone else is paying for it, right? Because I can't imagine just doling that money out over and over and over and not caring enough to do well. So I didn't say that. I think I did say that on record. Okay. Okay. So here we go. So let's talk. So is there any other? Talk to me individually if there's any concern about. Do you think that would be a wise departmental decision for us to require that? Again, if you're making an 80 or better, it's optional. If you are making less than an 80, we really recommend. In fact, we're going to require that you show up for tutoring once a week, and we'll make lots of opportunities for that to happen. But we really think you need that extra help, and we're going to expect you to be there. Otherwise, why are you here? We'll drop you from the course. We'll see. We'll see. I don't like being mean, but I want to see people be successful. Right? And we got to spend some time. Okay, let's talk about the senses. And it's a crazy picture I took. Now, if I asked you, or if I asked my kindergartner, what are the senses? We were probably taught there were 5 senses. And what are they? Smell, taste, touch, vision, and hearing, right? So there's those 5 senses that we've been taught about. I want you to consider that the 6th sense would be balance, equilibrium, your sense of balance. It's not something we think about or talk about very much, and there are books out there that suggest that the 6th sense is, ooh, that weird stuff, right? But the 6th sense for the anatomy class is going to be your balance, your equilibrium. And I'll come back and talk about that in a few minutes. In fact, next lecture on Thursday, I hope you'll come, because I'll be sharing with you a little bit more information about balance. It's kind of my favorite topic, and you'll understand why after you hear my talk on Thursday. So let's talk about the senses. You just told me there are 5 senses, and I told you, hey, I also want you to include your 6th sense, your balance, in this conversation. Now, are you aware of all of your senses at any one time? Are you consciously aware? I mean, most of your sensory information coming in through your sensory neurons and going up to your brain or toward your brain never actually reaches your consciousness. Most of those signals coming in about the temperature in the room, lights and smells and sounds, most of that really gets filtered out by your brainstem. And that information only goes to your spinal cord, or maybe goes up to your pons or your medulla oblongata, or your cerebellum, but it doesn't reach through our thalamus and reach to our cerebral cortex. We're only aware of a very small percentage. Now, if I say to you, think for a moment about odor, all of a sudden you'll become consciously aware and try to sniff out something. If I say to you for a moment, think about things touching your body, suddenly now you'll be aware of your watch or your glasses or your earrings maybe. But you wouldn't have been thinking about it nor been conscious of it until I reminded you of that. Okay, so most of our senses don't reach our consciousness. And by consciousness, again, I mean our cerebrum, our cortex. Now, in order for us to receive our senses, there has to be a receptor, right? There has to be a way that we receive this information into our body. And we're going to break these receptors into two classes. One will be your general senses, and then there'll be your special senses. General sense is basically touch. It's going to be something that's over your entire body that you can sense. You can sense touch, pain, temperature, pressure, vibration. Those are things that we can sense all over our body. We're going to call that a general sense, or all the general senses. And then we're going to deal with the special senses. The special senses are only coming from the head. And there are 5 special senses. Now, as I go through these special senses, I also want you to be painfully aware of the cranial nerves involved. You don't get to forget your cranial nerves. Maybe you didn't quite learn them, but you're going to keep hearing about them. So as I go through this list on the bottom here, gustation, that's a fancy word for taste. And I want you to right now, we'll do this again in a moment, but what cranial nerves are involved with taste? 9 and 7. 7 was the front of the tongue, 9 was the back of the tongue. Remember this? Okay. Now, how about olfaction? Fancy word for smell. The olfactory nerve, that was number 1. It's okay. Vision. That's the optic nerve. That was number 2. Equilibrium and audition hearing. Both 8. Very good. Your vestibular cochlear nerve coming off the ear. So again, your special senses are all coming in through your cranial nerves housed in your head. Now, receptors that pick up these senses come in different types. Some receptors are very, very simple, literally just bare dendrites hanging out in your skin. And other receptors are built into very complex organs like your eye. Your eye is really just a very complex organ to allow you to have the sensation of vision. So we've got very simple to very complex receptors. We also have receptors that are measuring the inside of our body, your internal environment, and you have receptors that are picking up external signals. So we have both external and internal signals or receptors. And again, we are only aware of a small fraction of these sensations. Now this slide I'm going to read to you, then I'm going to describe it to you. I think you'll find the description kind of awkward at first, and then it'll make sense to you after a moment. So, bullet number 2. There is an inverse relationship between the size of a receptive field and your ability to identify exactly where that stimulus was located. Keep going. Number 3, if the receptive field is small, then you will be able to tell with great accuracy exactly where that stimulus is on your body. Whereas if the field is large or broad, then you're going to have less ability to tell exactly where it came from. So for this, I need a guinea pig. I'll use you. Are you available?
Sure.
I'm going to have you close your eyes, and I need both hands available, and I need your hands. So I'm going to just touch lightly with a pen, and I'm going to touch a part of her body, and then she's going to, with her eyes closed, with her other hand, touch where I touched her. Bingo, right on top, right on her finger. And the palm, same thing. She's going to be very, very accurate. But I'm going to go to your forearm a little bit. Give me a little bit of your arm. A little bit more, if you can. Okay. Now, pretty good. She's off by an inch. Off by an inch. Pretty close. Okay, off by a good inch. So what's going on here? Think about that little strip of man we saw on the sensory strip. Remember that guy? And he had a big part of his body that was his mouth and his face, and a big part for his hand, but not much for his leg or other parts of his body. That means that there are more sensory neurons located in your hand and in your face than any place else in your body. If I touch your fingers, there are a lot of nerve endings there. And so because there are so many, I know exactly where it is. There's a high density of them, and we would say that each receptor has a very limited receptive field. Whereas on your forearm, even worse on your thigh or on your back, I can touch you and you may be off by 4 inches, 6 inches, because there's very few sensory neurons covering the sensation on your back. So if I touch there, you have an idea where I am, but you don't know exactly where I am. Okay, so this is the idea of receptive field, and this is a fun thing to play, right? So just ask permission first, but play this with the kids, play this with your friends, but just kind of blindfold them or have them close their eyes, touch them lightly in one place, and you'll be amazed. On the hands, they'll know exactly where that is. On the forearm or on the shoulder, on the leg, they'll be way off oftentimes because this idea of receptive field. Does that make sense? Okay, so read this again. There's an inverse relationship between the size of the field and your ability to localize exactly where that stimulus is coming from. A few more concepts about receptors. Some receptors are on the top considered tonic. Some receptors on the bottom are considered phasic. I'll start with the bottom one. Phasic receptors are ones that, quote, phase out. These are the receptors, when you put on your glasses this morning or put on your watch, at the moment you put it on, you were aware at that moment that you had just put something on your body, but within a few seconds, you've forgotten that it's there, because those touch receptors toward the surface of your skin are phasic. They phase out. This picture is basically showing you that here's the stimulus, and here's the nerve impulse. We'll talk about that more next semester. And then after a while, it kind of just fizzles out. It just kind of stops sending a signal. Even though it's still there, right? The watch is still on my arm. It's still triggering. There's still a stimulus, but it kind of phases out, kind of gives up, ignores it. That would be also true of your smell, usually. Someone walks in the room, they have cologne on, you smell it for a couple of moments, but then you forget about it. It's no longer part of your thinking, it's phased out, phasic receptors. Other receptors, though, are tonic at the top. They never stop sending their signal. This would be sight, right? Our sight is continuous. It doesn't kind of start and stop. Balance is tonic. It doesn't stop. Pain is tonic. Nothing's worse than pain, right? Pebble in your shoe, you're not going to suddenly forget about it. That pain signal is going to keep on being sent to you no matter how much you wish it to go away. So some receptors are tonic, some receptors are phasic. We also want to classify receptors based upon their location. So again, general sense, right? The general senses were like touch and vibration and pain and hot and cold. Those are distributed throughout your skin. They're also distributed across your organs, and I'll talk about that in a moment. And then those special senses, again, they were all coming from the cranial nerves, all coming from the head. Now, every receptor can be described using 3 different terminologies. We've always got the origin. Where does it begin in your body? The stimulus origin. And then there's the distribution of it. So where is it? And then how many are there? Where is it distributed? And thirdly, what's the modality? That's a fancy word for saying what triggers that receptor. Is it light? Is it sound? Is it a chemical? Is it touch? What is it that causes that signal to be triggered? So we'll start off with origin. Well, there's 3 different basic locations for these receptors in your body. Some receptors are called exteroceptors. Some are interoceptors, some are proprioceptors. This is not difficult to imagine. Your exteroceptors are found on the outside. They are picking up external signals. Your special, vision, smell, all of that would be exteroceptors. Interoceptors would be picking up internal, visceral responses. Is your stomach full? Is your bladder empty? Is your heart rate high? Is your breathing rate high or low? So those kinds of signals, those visceral sensory signals, would be described as interoceptors. And then we've seen that word proprioception before. What was proprioception? Knowing your own position in space. So those receptors are located where? What tells you your body's position? Those proprioceptors are connected to your muscles, joints, right? Muscles and joints and tendons. So muscles, joints, and tendons have receptors on them, and they're telling you, is your muscle relaxed? Is it tensed? Is your joint bent or straight? It's giving you all that information. That's proprioception. So I've got a slide for each of these, right? Exteroreceptors, again, picking up signals from the external environment. These are going to be found all over your skin. They're also going to be found on your mucous membranes. So inside your oral cavity, places like that. Number 2, interoceptors. Again, this isn't difficult. These are also called the visceroceptors because they're picking up signals from inside your body. These are usually stretch receptors. In other words, my bladder is stretched, my stomach is stretched or empty, my blood vessels are stretched or not. Is my lung being stretched? Is my heart being stretched? So many of the internal signals have to do with stretch, but there's also some chemical changes. Remember I talked about, or temperature changes. If I drink a Slurpee really fast and I trigger my vagus nerve in my stomach, I'm going to know it in my brain because I'm going to get a little brain freeze. So that'd be a temperature change, wouldn't it? Internally that's being detected. And there's also possible for some chemical changes inside your body as well. Finally, we have those proprioceptors. And again, as we've already said, they're located on your muscles, tendons, and joints. These are going to help you know, with your eyes closed, where you are. What position are you sitting? Are you standing? Is your arm bent or not? You know all that through the collective information coming from these proprioceptors. And then we have the distribution. Where are they found? Again, the general senses are on the body. You're going to be able to tell chemicals, right? You could burn yourself with a chemical. You could have a different temperature change. You could have something pushing on you. Pressure changes. You can pick up touch. You can tell if something is silky or not by fine touch. You can pick up difference in proprioception on the outside as well as pressure. So if I'm pushing or stretching on something, those are all the general senses. Versus, as we've already said, your visceral senses on the inside, same idea, usually stretch, sometimes chemicals, sometimes temperature. We've also got the special senses, and I've already said this, let's make sure that you've got your cranial nerves written down if you didn't do that already. So these are all coming from your head. Gustation, perception of taste, cranial nerves, what again? 7 and 9. Olfaction, cranial nerve number 1. Vision, number 2. Equilibrium and hearing, both number 8. Good. Then finally, what triggers that signal? If it's a chemoreceptor, right, then that receptor will be triggered by a chemical. Could be like glucose tasting sweet or sodium tasting salty. Thermoreceptors would respond to what? These make sense. Thermoreceptors are going to respond to temperature. Photoreceptors are going to respond to light, right? But also, think about this, not only do we perceive light through our optic nerve, but we also perceive colors, but we also perceive movement. Right? If you see a shadow or something moving, that's all part also of your optics and of your vision. Then we've got mechanoreceptors. Something is mechanically being pushed or shoved or touched. So these are going to be your stretch and touch receptors. Underneath this, there is a special category called a baroreceptor. When you hear baro, what comes to mind? I'm hearing bariatric surgery, a change in weight, or barometer, a change in pressure. Baroreceptors are specifically about pressure. So baroreceptors are a type of mechanoreceptor, or touch, or something like that's involved. And then the one that you're probably not familiar with is nociceptors. Noci, N-O-C-I, is meant to be sort of an abbreviation of noxious. These are painful things. Something that's noxious is painful or overwhelming. So nociceptors are receptors that respond to pain. Have any of you ever, well, we've all had pain of some sort, but have you ever had some habanero peppers or some hot sauce? Right? That can be painful. Habaneros do not trigger taste receptors. They trigger nociceptors. So that pain is real. It's not just a flavor enhancement or distraction. It is truly triggering pain receptors if you bite into a habanero or drink a little bit too much of that hot sauce. Okay, so let's take a look at this picture, and we've already talked about this a little bit. What we're seeing here is just normal old nervous system wiring. Here, though, we're looking at an internal structure. We recognize there's the cecum and the appendix hanging off from it. If the cecum or appendix, it has nerve endings, right? These would be visceral sensory neurons coming in, and they come into some spinal nerve. There they go into the spinal cord. We know the story. But also, that would be a visceral sensory neuron, right? But there are also going to be somatic sensory neurons traveling in from some area of the skin. And that information is also traveling into the same nerve. Sometimes the brain gets confused and it says, I don't know if that signal is coming in from the skin or if that signal is coming in from the internal organ, but I sense something's not right. So there's a pain signal that's generated. When a person's having appendicitis, they're going to have abdominal pain. It's going to be like the pain is on the outside of the body. It's all about a misinterpretation. So the brain is kind of confused because the signals are coming in on the same spinal nerve. Is that signal coming in about visceral pain, or is that signal coming in as somatic pain? We talked about one example earlier. Remember we talked about how if someone's having a heart attack, not always, but oftentimes they'll complain about radiating pain down the left shoulder and down the left pectoral region, right, left arm. That's because the heart is innervated internally by T1 through T5. And if you went back and looked at the dermatome map, remember those little segments of skin, you would see that these are T1 through T5 that go down the arm and are across the top of the pectoral region. So again, the brain's confused. Is it the heart that's in trouble? Is it the skin that's sending me a signal? I don't know, but you perceive the pain on the arm, even though it's the heart that's actually in trouble. Same thing is true if you've ever had a kidney infection. You might know that the pain is going to be across your lower back. Okay, so the kidneys are in trouble, but you perceive it as a backache. Or ovarian pain is showing here. That kind of makes sense because the ovaries, two ovaries. The appendix, way over here, right? This one's really interesting. If you're having gallbladder or liver issues, you might feel the pain here. That's kind of where the anatomical organs are. But you might also feel pain in the shoulder, the right shoulder. So again, that's that referred pain issue. So some people having a gallbladder attack will actually feel pain not only in their gut, but they'll feel it in their shoulder.
What about your
Spleen, I don't have that one on here, and I can't remember exactly where that pain is, but it's going to be back, I know.
back?
Yeah.
So if you're having pain in your stomach,
It could be any number of things, but my point is that some of these, I mean, if you're having lower back pain, it may be because you have a kidney infection, not because you've been hit in the back or done anything to pull muscles in the back. You may actually have an internal organ issue like kidney infection, or, now the stomach makes sense, right? The stomach's kind of right where it is anatomically, but you might actually feel that pain on your back. Not only on the front, but you might also feel it in the back. So again, it's all, we didn't go back and figure out exactly where all the organs are innervating, right? But remember that every internal organ is sending signals into one of your spinal nerves. And each part of your body is assigned to those dermatome regions. And so this is that mixing and misinterpretation of those signals. I don't know if that answered your question or not, but just don't memorize this table, but just have an appreciation of this idea of referred pain. So that's my little introduction to receptors. They all have a distribution. They're located inside and outside, and they respond to different modalities, different chemicals, different sensations. We're going to spend a few minutes talking about touch, then we'll talk even less about smell and taste, and we'll finish up today with vision. Then on Thursday, I'll continue the conversation on hearing and balance, and we'll move on to a little bit about hormones and the endocrine system. And depending upon how things go Thursday, I may or may not get to starting a little bit about the blood. We'll see, okay, how things go on Thursday. But remember, everything I'm talking about this week is pretty much not on an exam, just on your take-home exam, so a little bit more relaxed. So we'll start with touch. Tactile, fancy word for touch. These are the most numerous type of receptors in your body. They are going to be mechanoreceptors. They're going to respond to some sort of mechanical interruption, touching, pushing, vibration, pressure, something like that. Most of these tactile receptors will be located in the dermis. We'll see a couple, though, that do come up into the epidermis. And they're also located down deeper in the hypodermal or the subcutaneous layer. These tactile receptors can be very simple. By that I mean that it's kind of just a raw dendrite sticking out, or they can be more complex. And by that I mean that they're wrapped with some connective tissue layers. And you'll see the difference as I go through this picture in a moment. Now, tactile receptors, we'll start with the simple ones. They're also called the unencapsulated. They're not encapsulated in a connective tissue structure. This is going to include your free nerve endings. They're basically little dendrites sitting up in your epidermis. Picture's coming. Number two, the root hair plexuses. Now you know that a plexus is a weaving group of nerves. Every single hair on your body has at the bottom of that hair shaft a set of nerves around it, which is why it hurts to pull a hair. And it's also why if you just barely touch your skin and you just bend those little hairs on your body, you are aware of that touch because it's connected directly to a nerve. So that's the root hair plexus. And then there are also tactile discs. These were described by a name called Merkel, so they're called Merkel discs as well. And they're going to help you feel fine touch. So those are three simpler ones, the unencapsulated. And then there are also the encapsulated. There are four of these. The first, Krauss bulbs. These are going to be found only in your mucous membranes, and they're going to detect light pressure. There are these things called lamellated corpuscles. They were described by a name called Pacinian, so they're called Pacinian corpuscles as well. They're going to detect deep pressure. Now, I want you to notice something about that word. Cross off the last three letters of lamellated, and what do you have? Lamella. Now you're thinking layers, like the layers of an osteon. When you see this receptor, you're going to see why it's called a lamellated structure. Then there are the Ruffini corpuscles. They are located in the dermis. What's special about them is that they do not adapt. Another way of saying that is that they are tonic. They are not going to phase out. They're going to keep on sending that signal, and they're usually responding to continuous pressure, like someone's pushing down on you and it's not letting go, or twisting your skin, some sort of distortion of the skin. They're not going to stop telling you that. Then finally, there are the tactile corpuscles. A guy named Meissner came up with these, so they're Meissner corpuscles, and what's unique about these is that they are phasic, so they're going to phase out. These are the kind of receptors that are going to forget that you have your watch on, forget that you have on your glasses or your rings. So here's a picture, a classic picture of a slice of skin, and in this picture are all 7 different types of nerve endings for touch. You don't have to memorize this. In previous semesters, you had to learn this and know it perfectly for a test. This is all going to be open book, so you're not stressed out too much. Let me just go through these in no particular order. I will start out, however, with the 3 that were the unencapsulated, the more simple kinds of receptors. If you look carefully in this picture, you're going to see that over here there are some nerve endings that are kind of permeating up into the epidermis. Let's remind ourselves of what we have here. My finger is drawing along the what? Basement membrane. Remember this? And the basement membrane is going to have on top of it one layer of cells called the stratum basale. And then there will be multiple layers of the stratum spinosum. then the stratum granulosum, and then the stratum corneum. Okay, we know this is thin skin because I see hair. All right, this isn't my palm, so that would be hairless. So this is thin skin. I don't have the stratum lucidum. Remember that little conversation? Now, where are these little nerve endings? These are little free dendrites up into the epidermis. Have you ever had a paper cut? Man, they hurt. Sometimes they bleed, sometimes they don't bleed. Well, if you have a paper cut that bleeds, what do you know? You've gone down into the dermis, because that's where the blood vessels are. Right? Makes sense. It hurts, but it bleeds. But if you ever had a sliver or a paper cut that didn't bleed, but man, it hurts, what'd you do? You only sliced deep enough. You didn't go all the way down to the blood flow, but you did at least cut into some of these nerve endings. Man, those hurt. Right? So that'd be some of those free nerve endings at the very surface of your skin. Also, in this picture, down here at the bottom of the root of the hair, see that group of nerves? That's the root hair plexus. It's why it hurts to pull a hair, and it's why when you barely touch a hair, you're aware of it because those nerves are picking up the signal. Then finally, there are these guys over here on the far left. These are the Merkel discs, and you see that they're little nerve endings. They only come up as far as the stratum basale. Again, these are going to pick up light touch. It makes sense. They're toward the surface, so you don't have to push down very hard for these receptors to perceive silky versus rough. Those are the three simpler ones. The four encapsulated nerve endings are here. Start here. This is the Meissner. Looks to me like a little arrowhead, little pointy thing. It's up toward the surface of the skin, up by the top of the dermis. And this is also going to help you determine touch. These were the ones that I told you that phased out. They were phasic. So this is your watch going on and forgetting about it, your ring going on and forgetting about it. Right here, this little guy, that's a Krauss bulb. Those are the ones found in the mucous membranes for light vibrational pressure. What's this guy? It's way down there, but it looks like an onion, doesn't it? Has many, many layers. That is your lamellated or Pacinian corpuscle, so laminated or Pacinian. That's going to detect deep pressure. That makes sense, though, because where is it? It's deep down in your dermis. For that to get activated, you've got to be pushing pretty hard on your skin to activate that particular receptor. Then finally, I wrote over it, there's this little blimpy looking receptor right there. That's the Ruffini corpuscle. And what was unique about that one is it does not adapt. So if you're pushing on your skin or you're twisting your skin or distorting your skin, it's not going to stop sending the signal. So Meissner's were phasic, right? They phased out. Ruffini's were tonic. They don't phase out. So that's really what I want you to know about touch and these different kinds of receptors. Now the next 5 or 6 pictures are just individual blow-ups of each of these. I'm not going to go through these individually. You can review that. Definitely when you're taking your take-home exam, you want to make sure you have your book nearby, but you also want to make sure you have your notes nearby. And because you're also dealing with the eye and the ear on that take-home exam, I'd make sure I had my lab manual in front of me, because you might have structures about the eye or the ear that you have labeled already and are very familiar with in your lab manual. So when you're doing your take-home exam, have your book, have your notes from lecture, and have your lab stuff with you and nearby so that you have all of those resources. And of course, you still have Dr. Google. He's just a few keystrokes away. One more thing about touch is this thing about phantom pain. Have you heard about this? A person can have lost a limb and still sense pain or sensation from that particular area. So this person has had at least one amputation below the knee, and it's possible that they're still going to feel as if they have that limb. It's just, again, it's a screw up, it's a misinterpretation of the nervous system. And the brain may perceive, they may reach down to scratch the limb that's missing. Now that could be kind of humorous in the right context. But it's also possible that there's pain, constant pain coming from these limbs. And so that can be a real problem. And sometimes they have to go in and actually do a nerve block, even though it's not there. They have to go in and do a nerve block on that part of the limb, so that the nervous system's not perceiving that pain. It's really odd, right? It's not even there, but yet the brain is perceiving pain from that amputated organ or that amputated structure. I think it can. I think it's like anything else. I don't know if they would be considered tonic or phasic, but I've heard that for those who have the painful sensations, it can be really overwhelming. So that suggests to me that it doesn't just go away, that they actually have to do something medical or surgical to correct it. That's all I'm going to share with you about about touch. So that's kind of an overview of the different kinds of touch receptors. And you just know that it's kind of complicated. There's lots of different kinds of receptors, 7 different kinds of receptors that give you the totality of your touch experiences. That would be general senses. Everything from this point on is going to be your special senses. And we're going to start off with a couple of slides about taste or your sense of gustation. Now, we know this, right? Your taste buds are located on your tongue. Duh. They're located on these bumpy structures of your tongue called papillae. Now, there's that word again. We've been seeing different variations of this word. In the skin, back in chapter 4, we talked about the papillary layer of the dermis. That was the up and down, peaks and valley layer. In the heart, we're going to talk about the papillary muscles within the heart. In the digestive system, we talked about the duodenal papilla, little structure where the bile and pancreatic juices drop in. And we'll hear a lot of other papillary-like structures this semester. Papilla, vocab, remember, it means nipple-shaped. So these little bumps, these little bump-like structures on the tongue, and they come in 4 different types of bumps. You're not going to memorize them. It's just kind of an OG whiz kind of thing. But they've actually described these 4 different kinds of bumps on the tongue. They're a filiform, which have nothing to do with taste. They're just kind of there as a create friction, so you can roll food around in your tongue and make that bolus of food when you're swallowing it. Cats have, right, kind of a rough tongue. So a rough tongue idea. Then there's fungiform. They're kind of more on the side of the tongue. You're not going to memorize this. There's a few taste buds in there. Most of your taste buds are actually back toward the back of your mouth. These are your valate or circumvallate papillae. And then there are the foliates as well, which really have nothing to do with taste in adults. Look at the picture, not to memorize. There's your tongue. We're going way back, right? So you have to go way, way back to see any of this. But along the surface of your tongue, you would have these little bump-like structures. And here's one, right? And here's another one. And down in those grooves next to those bumps, these little circles represent taste buds. So you can see those little taste buds are down inside those little grooves. So chemicals are going to have to migrate down into those little grooves and activate these taste buds. Here's what it looks like. So we've zoomed in to one of these little tiny, tiny taste buds, and this is what we would see in a cartoon. At the surface of the tongue up here, there'd be little pores, little openings for the chemicals to go down. Down here are the nerve endings. You see the yellow. Those nerve endings are going where? They're all going to go where? To what cranial nerve? 9 or 7, depending upon where we are in the tongue, right? 7 or 9. And you've got all these little, looks like a little balloon or a little punching ball to me. You've got little cells in here. These are your taste cells. Now, it turns out that these cells are being replaced all the time. They're replaced every 7 to 10 days. And in fact, after age 50, taste starts to diminish. And so that's when you start seeing people pouring more salt on their food, because their sense of taste does begin to decline a little bit with age. This is a good time to talk about a connection too with chemotherapy. I forgot to mention this morning. But if a person's having chemotherapy, right, cancer treatment, what are some of the complaints? People who are undergoing chemo treatment.
They get a weird taste.
They get a metallic taste. Nothing tastes right. Not to mention they're getting diarrhea. They got upset stomach, a lot of nausea. Their hair falls out. What are all of those things connected to? Hair falling out, nausea, diarrhea, and taste issues No. What, all of those are, what's the chemo doing? It's trying to kill off
That's
cancer, tumor cells. And one of the characteristics of tumors is that they're fast growing. So we're going after fast growing cells. We're giving them poison, basically. We're assuming that the rat's going to eat it, right? That the tumor is going to eat this poison and get killed off. But what other cells are growing fast? Gut lining, hair follicles, and taste buds. Right? They're all regenerating very quickly, so they too are getting killed off. Now they'll regenerate, most of them. The hair will come back, the sense of taste will come back, they'll stop having diarrhea, they'll stop having nausea when the medication goes away. Right? Hopefully we kill the tumor cell in the meanwhile.
A little bit off topic when you brought it up. So would platelets be affected or not?
Absolutely. That's the other one I didn't say, is that these patients are also very anemic. They have low red counts. They become immunocompromised because they have low white cells, and they don't heal well, they bleed easily, and they have clotting problems. So absolutely, all the blood cells are also affected by chemo, right? Feeling weak, low red cells, low oxygen, low immune system, low white cells, and it compromised clotting and healing as well. Platelets, so absolutely. Those are all also very fast-growing cells. Okay, so I just mentioned that as a aside, that 7 to 10 day, very short lifespan, and that's why they are affected so much by chemo agents, for example. Now, in the nervous system, I described to you a pathway. And I said a pathway was basically a tract and a nucleus, and that was a little bit vague for some people, I think. This picture would be that of the taste pathway. What that simply means is this. Information is coming in from the taste buds. Those taste buds are located on the tongue. If it's in the front 2/3 of the tongue, that information is going to go up and be part of cranial nerve number 7. If it's in the back third of the tongue, it's going to innervate 9. Now, where does that information go? It then goes into the brain, right? It goes into the brain. It goes into, come on. I have a little reconnect message here. Okay, and we see that that signal is going to come in cranial nerve 7 and 9. Now, where is it going? It's going to go up to your thalamus. Remember, everything goes up through your thalamus, and it's going to go up into your cortex, into your cerebrum, and we would refer to this area as your gustatory cortex. Now, what does that mean? It's the part of your cerebrum, the cortical region, the outside edge, that's receiving information about taste. It is possible to have brain damage and lose your sense of taste. Right? If you knocked out that portion of the brain, that little gustatory cortex, and what lobe are we in? Here's your temporal lobe. And I'm gonna kind of cheat here and make you think, here's frontal lobe. That's gonna be parietal lobe, because what do we know about the parietal lobe? Senses, right? Sensations. So this gustatory cortex is going to be a part of the parietal lobe. Now I want to say one more thing that may help you make connections. I haven't introduced this before, but I feel like I need to say it right now. In your thalamus, there's a portion of your thalamus that also is involved with your emotions. Can taste change your emotions? Chocolate? Right? Grandma's cooking? Blah. Right? So we know that our mood and our overall mood can be influenced by taste, can it not? Okay? Many different things can influence our mood. So what I want you to start thinking about is that, yeah, when you're tasting, not only are you simply tasting for the sense of tasting, but you're also informing your emotional centers of your brain. And you're going to see that that's true of vision and smell and taste. All of these senses can reprogram or influence our well-being and can make us feel more relaxed or make us feel more anxious. They can change our emotional status. So, I'm getting a little bit off topic. We'll talk a lot more about this in 106, but I want you to realize that your senses are not just coming into your brain for the purpose of sensing something like seeing or hearing, but some of this information is also taking a little side trip and informing the part of your brain that's involved with emotions and your well-being and your sense of safety and all of those things in your brain. Now, if I asked my 7-year-old what different kinds of tastes are there, and maybe you learned the same 4. Okay, I learned that there were 4 taste sensations in elementary and middle school. And, we learned about them, right? Salty, sweet, bitter, and sour. You agree? But about 20 years ago or so, there was a Japanese group that described a 5th taste sensation that you'll hear about in soy sauce commercials called umami. Now, umami from the Japanese apparently translates as something like meaty or savory. So there's actually 5 different taste sensations that our tongue can play with. So number 1, salty. You tell me, these are chemoreceptors, right? They pick up chemicals in our food. What chemical is going to give you the sense of salty? Just sodium. The chloride, that's part of your table salt, right? Sodium chloride. Anything with sodium in it is going to give you a sense of salty. Sodium is the trigger for saltiness. What about sweet? Simple sugars like glucose and fructose, but glucose we know. Sour. When I say sour, what comes to mind? Warheads. Lemon, right? And lemon is very acidic. So what's triggering your sense of sour is going to be H+. Remember we talked about more acidic things have more H+. So H+ is going to give you a sense of acidity or sour. Now, bitter is not something we walk around and choose to eat very often. And I don't have a good molecule for you here. Bitter things like bitter herbs or coffee has some bitterness to it. Some wines can have bitterness to it. Seltzer water can be very bitter. It's not something we would normally choose to eat or drink. But bitter, there's a molecule called quinine. I think I'm spelling it right. It's not something I think about too much. But quinine is one molecule that can trigger that bitter response, not something we're familiar with. The last one, though, is interesting. When you walk into many Oriental restaurants, Chinese buffets, they may say with pride above their buffet, we do not use MSG. MSG is a flavor enhancer, and it actually triggers this umami sense. So the actual trigger here is glutamate. Now, glutamate is one of the 20 amino acids, so it makes sense that it's going to be meaty, protein kind of thing. But if you add MSG to something, you're not only adding savory, whatever that is, but you're also adding a lot of sodium, because monosodium glutamate is also salty. So if you're having soy sauce, right, there's a lot of sodium in it, it's salty, but it's also adding this flavor additive, however you want to describe it, as umami. So those are the 5 different flavors or tastes that we're able to differentiate. There's one food group, I call it a food group, one additive, one condiment that kind of triggers all 5 of these and has been proposed as to why Americans are kind of addicted to it. What food group do you think triggers all 5 of these senses? And so we pour it on our food.
Soy sauce?
Ketchup. I've heard it said that ketchup is such a popular food item because it has a little sweet, a little salty, a little sour, a little bitter, and, oh, by the way, it has some glutamate in it. It's got a little umami. So it triggers all of our taste sensations. And Heinz and the Doles and whoever else are all very happy. Heinz and Hunt's, right? Very happy that ketchup just happens to hit all those flavor sensations at the same time.
One of them's a lot sweeter than the other.
Yeah, there's differences, right? But ketchup does kind of have a little bit of each of those, and maybe that's why we pour it on a lot of things. Okay, that's all I'm going to talk to you about taste. It's not too much. I'm going to go on and tell you even less about smell, your sense of olfaction. Any questions on taste? Pretty straightforward, right? 7 to 9, make sure you know your cranial nerves. Olfaction, sense of smell. We know this is the olfactory nerve. We know this is nerve number 1. And just like any other nerve, there are going to be other cells that are going to protect it. This is actually the only nerve that is exposed to the outside world. The olfactory nerve is sticking out into your nasal mucosa. Part of it's sticking out into your nasal passages. So it actually is exposed to the outside world. And so there's some other cells that support it in there. Don't worry about those names of those cells. It turns out that there's about 50 or 60 distinct smells that we can detect as humans. We've got 5 taste molecules, flavors, but we have about 50 to 60 different distinct smells. And from those 50 or 60 different odors, we're able to now, imagine thousands of different combinations. Now, some of our animal friends have a more distinct odor or sense of odor. So dogs, for example, have more different types of receptors available to them, so their sense of smell is more critical. So taking a look at this, here's your brain over here. And your first cranial nerve comes all the way up underneath the frontal lobe. Now right there, if you think back to cranial anatomy, there was something sticking up into the frontal bone that was actually part of the ethmoid bone that kind of stuck up, and we called that bone marking the crista galli. On each side of that little crista galli, if you look at a skull again, you will notice there are little perforations, little openings, little holes on either side of it. Those little openings are the openings, here we go, in this picture, here's your big olfactory nerve, and those little holes on either side of the crista galli allow for the nerve to come down, and then sticking outside on your mucous membrane of your nose, they call them olfactory hairs. Now that's a really kind of misnomer. It's not like hair on your body or hair on your head, but they're actually referring to little branches of dendrites. So these little receivers of information are sticking down into your mucous membranes and are detecting these 50 or 60 different distinct odors. This is why if you are stuffed up, you can't smell very well, because you're basically covering up those dendrites. The molecules have to be able to dissolve in and permeate through that mucous membrane. Now, some molecules can permeate through that mucous membrane better, or that mucus gunk. So some molecules like menthol or minty things or eucalyptus kind of things, those odors are able to permeate through the mucus. So we smell those things better when we've got a stuffed up nose. But it makes sense. Yeah, vapor rub, right? It has all those chemicals that are actually able to permeate right through that gunk, right? That would be cluttering up the taste receptor, or the smell receptors. So again, all this information is going to travel up into the brain and travel to, or through, the olfactory nerve, right? Number 1. That's all I'm going to talk about for smell. It's probably the only special sense that we can live without tremendous change of our life. Right? My wife's grandmother had no sense of smell. She never had one. They don't understand. She was just born that way, and they finally figured out why her cooking was so horrible. But honestly, she could burn food and didn't know the difference. And smell and taste are kind of all connected in some ways. So, bless her heart, we just didn't eat there very often. I mean, we would eat, be very polite, and then stop at Blimpie's on the way home. It just was not very satisfying. And she didn't salt or season her food very well either, because she had none of this. She had some taste and some smell issues. It just was the way she was. But we can live without it, right? It may be a little awkward socially when you're serving food that has no flavor, or vice versa, or blazingly salty or something and nobody knows the difference. Last thing will be vision. And we'll talk the last 15, 20 minutes about vision. And that will get us through today. And then we'll pick up, like I said, with hearing and balance on Thursday. And finish up with the endocrine system also on Thursday. Now, when you're doing your take-home exam, like I said, make sure you have your lab book out. I will only ask you for the same structures of the eye that we knew in lab or know from lab. Same is true for the ear. So I'm not pulling out new structures. So make sure that you limit your, eye structures and you're thinking about this to the structures you've already been exposed to. So the eye really is just a really fancy receptor, right, for vision. And you got eyelashes and eyebrows and all those things, basically to prevent something from interfering with your vision, catching particles. You've got your tear glands keeping your lens clear, keeping your cornea clear. So all of those things really are just to maintain good vision. There's also a conjunctiva. Now that's one structure that I did not have you know from lab, but your entire eye is covered even outside the sclera. There's a very thin layer called the conjunctiva, and you may have had conjunctivitis, pink eye, right? So that in that very superficial layer of the eye can get infected with a virus or bacterium, and we spread it very easily, pink eye or conjunctivitis. But all of these things, right, are there pretty much to protect your sense of vision. When you look at a picture like this, don't worry about anything that wasn't already introduced. So don't worry about, the medial commissure, the fissures, and some of the things that are on here. Just as a reminder that the eye, this very complex thing, it's embedded in your head. You've got these eyelashes and eyelids and all these eyebrows. All that hair is there really to protect things from falling into and perhaps damaging your eye. You've got the lacrimal glands, your tear glands producing tears that wash across. Remember that we have the opening, that's the pupil. And the muscle of the eye, the colored muscle of the eye is the iris, remember this? And the outer white layer of the eye is the sclera. Also going over the iris and the pupil would be the cornea, right? The cornea allows light to go through. Oops. So here's an image. Just, again, ignore the things we haven't talked about before. But on this, let me point out a few things as a reminder. So, start off with some basic stuff. This is the optic nerve, right? Cranial nerve number 2 coming off the back of the eye. This picture shows us 3 layers of the eye. The inside layer would be the retina. The middle layer would be the choroid, and the outer layer would be the sclera. All right, so we've got that down, we know that. This big space back here, filled with a ball of jelly, that is the posterior cavity, right? And it's filled with that ball of jelly called the vitreous humor, right? That ball of jelly that we saw. Here's the lens. What's this muscle coming down from this view? That's the colored muscle. That would be the iris. And the opening from the iris would be the pupil. This would be the cornea. Okay. As I mentioned, you have lacrimal glands. These are located superior and lateral above your eye. They're producing tears. Those tears are now draining across your cornea, across your sclera, across your conjunctiva, and all of that tear is draining down into a lacrimal duct, which drains into your nasal passages. So you are constantly making tears, and you're constantly swallowing that fluid. So that's, you're always swallowing. Part of that is your tears always going across your eye. We don't think about it. Now, some individuals have dry eye syndrome. We've seen these commercials, right? People whose eye stops making these tears. Now, what would be the problem there? Every time you blinked, right, it could be a little bit rough and scratchy. Any kind of dust particle or something that got in your eye would not be washed away as quickly. Bacteria and virus could cling there and cause more infection. And the cornea is extremely sensitive. Has anyone ever scratched their cornea? I have not, but it's extremely painful. I mean, if you go to touch your eye, you know that that's not something you want to do. And so imagine you're scratching your eye just from particles and dust. So this would be a big problem. So you see that Restasis, some of the medications now, right? The doctor is telling the patient, oh, you have dry eye syndrome. Use this, and there's now something that's actually helping to improve the production of tears. Now, oftentimes, people who have dry eye syndrome also have dry mouth syndrome. They typically kind of go hand in hand. So they might have dry eye syndrome, but they might also have decreased salivary function. Now, what's that going to be a problem for? Digestion, eating, healing inside your mouth, because remember your saliva has growth factors and antimicrobial and antiviral factors in it. So if you don't have good salivary flow, bacteria are going to cling to your teeth longer, set up more cavities and more caries, more plaque production, overall just unpleasant, right? So these individuals that have dry eye, typically also dry mouth syndromes that go with it. And some of those symptoms are signs of some autoimmune diseases, which tend to hit women more than men. So most of those commercials are going to be dealing with women because that's going to be the larger audience. So we got these little lacrimal glands that come across, wipe out the eye, helping out to protect. Again, this is all about protecting the sense of vision, isn't it? These lacrimal glands. Let's take a look. I've got about 3 pictures of the eye just to remind us. It's been 2 or 3 weeks since we've seen the eye. This one's nice. It shows all 3 layers of the eye pulled apart. The sclera, the choroid, and the retina. It shows that big ball of jelly back here. That would be the vitreous humor found within the posterior cavity. We see the lens. The lens is attached to those suspensory ligaments. This is the iris coming down, the muscle. The pupil would be the opening. And then the cornea would be the covering over that. It's not labeled on this picture, but remember that the entire eye would also be coated by that very thin layer called the conjunctiva. What I want to point out here are the cavities or the spaces. And there's another picture coming up as well. But this space right here, the anterior chamber, is where I'm filling in right now. It is the space basically between your cornea and the iris. That area is filled with aqueous humor. That's the water that came squirting out at you when you dissected your eye. We didn't have aqueous humor on the list because it's kind of hard to label water. So that's that aqueous humor, watery kind of substance. Now behind the iris and kind of intermixed with the suspensory ligaments, that is the posterior chamber. Now that is also filled with the same watery aqueous humor. Remember that aqueous humor is different, right? All of this area where I've got my big arrow, everything in front of that is filled with aqueous humor. Everything behind that is filled with the vitreous humor, the glassy ball of jelly. So that's what I have here in words, what I just told you in picture. So we can divide the entire eye up into 2 big cavities, the anterior and the posterior. And that's where that big line I just drew. Everything in front of it was the anterior cavity, everything behind it was the posterior cavity. Here's where the confusion starts. That anterior cavity can then be further divided up into 2 smaller chambers. So I talked about the anterior chamber and the posterior chamber. They're both part of the anterior cavity. Just look at the words. It's not that difficult. But let me go back and show you that in the picture again. In other words, everything to the right of this would be the anterior cavity. The anterior cavity can then be broken up into 2 chambers. Posterior chamber, anterior chamber. Okay? So that's where the confusion comes. There's a posterior cavity and there's a posterior chamber. Look at that. Now, aqueous humor, why do we need it? The cornea, it's cells, right? We don't want to forget that the cornea is also cells, and the cornea needs, those cells need nutrients. They need oxygen just like any other cell in your body. Now, the cornea can get oxygen from the external environment, right? Where the cornea is out exposed to the environment, but the cells on the inside must get nourishment and oxygen from the fluid. So the aqueous humor is actually feeding the corneal cells from the inside. From the outside, the cells are being fed by the oxygen of the environment and the tears. They also have, nutrients in it for the cornea. You don't want to wear contact lenses that aren't designed for long term unless they are truly gas permeable. I know a lot of us are tempted, if you wear contact lenses, to save money and get that pair of daily wears to go 2 or 3 days. Well, those lenses are not designed to let gases go through, and your cornea needs oxygen, and so that can lead to some scratching and some irritation and some damage to the cornea. And most optometrists will tell you, give your eyes at least a day a week, right? If you're wearing contact lenses, let there be 1 day a week where you're not wearing them. Just go back to your backup glasses so that you're giving your eye a rest, letting your tears coat over your cornea, and not let any of that be interfered with. So that's your aqueous humor. Now, the vitreous humor is that big ball of jelly in the back. Now, you've seen it. You saw it pop out with the eye dissection. It's there for a couple of reasons. One, it's a big ball of jelly, and it is pushing on the big posterior cavity. Remember how thin the retina is? When you were dissecting the eye, do you remember the retina was a very thin layer? And in fact, most of you push the retina back to see that tapetum lucidum in your sheep's eye, that blue, beautiful Caribbean blue color that you saw. So the retina is a very, very thin layer. And that vitreous humor is helping to kind of push and keep that retina in position. But when you are looking at a 20-gallon aquarium and you stick your finger or a stick into the aquarium, it looks like the stick bends, right? So as light goes from air into water, there's some changing of the angles and refraction angles and things like that. That ball of gel is also helping to bend the light rays to the appropriate angle, so that as light comes through your cornea, hits your lens, and now goes back toward your retina, that vitreous humor, that ball of jelly, bends the light at the right angle to help it focus back on the retina. And if it were instead water, the light wouldn't bend appropriately to see well within that little ball of the
Are
eye.
you going to be talking about that like in 106 with like the cones and the rods?
The cones and the rods and all of that will come back in 106, or come to you in 106, absolutely. Right now in 105, we're just kind of talking about the phenomenon of seeing and the big picture in the structures, but in 106, we'll definitely talk about rods and cones and rhodopsin and how all that really, really works. Yeah. Okay, visual pathway. I showed you the taste pathway earlier. The visual pathway I alluded to before. So I want you to think about this. So light comes in to the eye. It's going to pass through the cornea. It's going to go through the pupil, hit the lens. The lens is then going to focus the light back on the retina. The retina is filled with photoreceptors that perceive the light. And that information is now going to travel down the optic nerve. We've seen it sticking out of the back of the eye. Now remember that a nerve is something that's outside of the brain. So as long as that nerve is outside the eye, or outside the brain, we're going to call it the optic nerve. But at some point, that nerve crosses over. If you turn over a brain, and I'll show you a picture in a moment, you're going to see that the optic nerve has what's called a chiasma. Now in the Greek, chi, that's the letter chi. It's an X, right? So chiasmata is referring to the crossing over of the optic nerve. So there's an optic chiasm crossing over, chi. And then once it crosses over, that optic nerve is going to penetrate into the brain. Well, we don't have nerves inside the brain. Instead, we have tracts. So we just changed the name. The optic nerve, once it's in the brain, becomes the optic tract. It's still that bundle of axons traveling together. And where are they heading? You know that the visual information, part of it, is going to go back to the occipital lobe. But, like I mentioned to you about smell or taste, information is also going to go by the thalamus. And the thalamus is going to be your area of well-being and emotional centers and anger centers of your brain. Could some images make you mellow? Some colors are soothing, some images are soothing, others are simulatory, others are angering, right, and can make you angry. So visual information also can easily influence your emotions. So it makes sense when we look at this picture of the pathway of the eye. Visual information, right, this is like coming from the left eye. The left eye has the blue axons, and we notice that the information from the eye on the left side is coming in. Not all of it, but some of it is crossing over, decussating at the chiasmata. And notice that some of those axons are going to go into the thalamus. Again, that's your areas of anger and well-being. So it's going to inform part of your brain about your well-being, your anger and emotions. And then the rest of that information is going to travel back to your occipital lobe, right? And we know that that's where your visual cortex is found. Remember I talked about the gustatory cortex? The gustatory cortex would be the region of your brain that perceives taste. The visual cortex is the part of your brain that perceives light. Okay, and that's back in your occipital lobe. Now I want you to notice something else here. Not all of the information decussates. Some of the information from the eye stays on the same side. So some of these axons you'll see stay ipsilateral, right? Stay on the same side of the brain. So your eye is crossing over, but not all of it is decussating. What does this mean? What if you lost part of your brain, right? You had damage to your brain or had a stroke in, let's say, your left occipital region. You no longer can perceive light. That would affect a little bit of both eyes, wouldn't it? But if the other side is working, what do you think about your vision? Can you still see? Right? And you're actually able to see from both eyes. Because both sides of your brain are receiving images from both sides of from both of your eyes. So it's actually a nice little protective thing. If every bit of our visual information from the right side crossed over to the left side and we lost one of your visual areas, you would lose complete vision in that eye, wouldn't you? There'd be nothing wrong with your eye, but the brain couldn't perceive the vision. So this is just another way that the brain is designed to protect our sight, this important sense. So if you damage part of your brain, you still have visual information being perceived from both eyes. Okay. Make sense? Kind of it's a bigger idea, but I wanted to share that with you. But clearly, visual information comes into our thalamus and informs our emotions as well. Just a couple more things about the eye structure. I don't want to beat this too hard because we've already talked about this. But when you saw your sheep eye, the lens was a little bit cloudy, kind of yellowish, wasn't clear. In you and me, that lens would be clear. In fact, the lens is made of a protein called crystalline. It's crystal clear. Right? Now, the eye can also be divided up. We talked about the 3 layers of the eye. There's another word for layer, and that word is tunic. And we're going to see this word pop up multiple times here in the next few weeks. So I want to introduce that word tunic to you. It's just another way of saying layer. So we can say the eye is made up of 3 layers or 3 tunics. And here's another way of describing those layers. Fibrous tunic, vascular tunic, and neural tunic. You know these by other names. The fibrous tunic, that's the outer layer, that is the sclera. But remember how tough it was? If you were the one with the scalpel in your hand when you were cutting the eye, you know how tough, fibrous that sclera is. So that's your tough, fibrous tunic. The middle layer, the choroid, is what you know it as, has a lot of blood vessels in it. It's extremely vascular. So the vascular layer, the vascular tunic, the choroid. And then the neural tunic, where the neurons for sight are, where the photoreceptors for light are, those are on the retina, also called the neural tunic. This next picture just kind of shows you those 3 layers, really, really clean. Here's your 3 layers, but I want you to notice something. The blue is your fibrous tunic. So here's the sclera, 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 sclera. But when we get in the front of the eye, Now it becomes the cornea. So the cornea and the sclera are continuous with one another, and together are the outer fibrous layer. Then the vascular layer, you're thinking choroid, 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 but watch, as I come around on the pink layer, the choroid actually becomes, or is continuous with, the muscle, the iris. So the iris is a continuation of the choroid in that middle layer, the vascular tunic. And then the retina only comes around halfway. There's nothing in the front of the eye that continues with the neurotunic. It's all just the retina. I've already said most of this, but I'll just say a couple of highlights and then we'll be done. So the cornea, well, I haven't said this. The cornea is transparent. Now again, in your cow eye, sheep's eye, whatever it was last month, that cornea was not very clear. It had been sitting in the pickle juice and the fixative for a while. It was kind of cloudy. It looked like a person was blind, right? A blind eye. But in your eye would be clear, your cornea, and that of a freshly killed animal would be clear. The cornea is avascular. That's a really good thing, right? That means what? That means that there's no blood vessels traveling through your cornea. You're not having to look through, or light's not having to move through blood vessels. That's good. It's avascular. But it also goes back to what I said earlier in that it's still cells, and it still needs to get oxygen and nutrients. So how does the cornea get its nutrients? Through the tears and through the aqueous humor. So you don't have blood flow going to the cornea, but it's still made of cells. The other thing, and I've already mentioned too, is that your cornea is highly innervated. So again, if you scratch your cornea, you know it. So while there's no blood vessels there are a lot of nerve endings there for pain. Then that middle layer, the vascular tunic, already mentioned how the choroid is continuous with the iris, the colored muscle of the eye. But let's think about that. Last time we talked about the sympathetic and the parasympathetic nervous system. And what happens to the iris? What happens to the pupil and to the iris when you're relaxed, parasympathetic? Shrinks, right? It contracts, right? Constricts. And what cranial nerve is involved with that constriction of the pupil? Ocular motor, number 3. Okay. Whereas when the bear is chasing you and you're scared, right? Dilation of the pupil, that's sympathetic. That's not a cranial nerve, right? That's not a cranial nerve. It's another nerve that does that. But in the parasympathetic nervous system, it's number 3 that does the relaxation of the eye. So here's just a picture of that. So when you're relaxed, right? When you're relaxed, parasympathetic, the eye is more constricted. And when you're scared, pupil's bigger, that is sympathetic, and that is not a cranial nerve that does that. And that, I believe, ladies and gentlemen, finishes up the eye. There's just one mention about the retina, and as you already mentioned, the retina is where you have the rods and cones, but we're going to deal with that next semester. So that finishes up the eye. We'll pick up with hearing and equilibrium on Thursday. I'll have a very personal story to share with you about that and struggles in my own life with equilibrium. So I hope you'll come and listen to part of that story. And then we'll also continue on with the endocrine system on Thursday. Okay, great.